So thank you for having me, and it's of course great seeing you again, Karen. For the students that are participating, or anybody who is online, if you have questions, I would love this to be somewhat informal, and I'm happy to answer any questions. Unfortunately, I have a hard time seeing the chat when you're presenting via Zoom, so if somebody posts a question in the chat and you're uncomfortable speaking, just maybe if somebody else notices a question in the chat, then just holler it out so I can answer it. I changed my title a little bit, and that's really, I'm going to highlight sort of the road of envy to the development of the metabolomics tools such as GMPS. And it's really an infrastructure that has come to be built for, by, and with the community. And so it was a really change in perspective. And realistically, so I'm a faculty now for about 15 years. So I started at UCSD in 2006. Never in my life would I have imagined when I started that I would be building a community analysis infrastructure that is now accessed, or at least last year was accessed 242,000 times a month. I actually just double checked while I was preparing for this presentation. Now we're getting 332,000 sessions a month to our platform. And so when I started my position, I would have never imagined that we would build an infrastructure that would be so widely used by the community. And so I'm going to give you a little bit of history, sort of taking a few years back. And we really needed to go back to 2007. So remember, I started in 2006 as a faculty member. I'd just done my postdoc, and I became very interested in some of these very simple questions. And basically, if you have a mass spec feature found in one study, let's say in a disease model, is that relevant to people? Or is a mass spec feature found by microbes or the host? And these are very simple questions. And if you're able to answer these kind of questions, you can start to really have profound implications in terms of how you would study and use mass spectrometry. And so this is everything I'm talking about would not be possible without this wonderful collaboration between Nuno Bandeira, who's a computer scientist. But at the time I met him, he was a graduate student at UCSD. And you'll see shortly why this was so critical. But I think most of you will agree that our ability to read these letters with relative ease from any biological sample has really transformed the way we do life sciences. So, and really the reason for this is because this allowed us to answer very simple questions. And so, are there any analogs? So mutations, SNPs, splice variants. That's something we could do by simply comparing one sequence to another sequence. We could answer the question, in what other data sets are these sequences found? This may seem very trivial. This is not trivial in mass spectrometry. And then once you find what data sets the sequence is found in, is you can ask the question is, okay, what's the context of that data? What's the geography of that data? Or what organisms is that sequence found in? Or maybe what disease states is that that sequence found in? And from that, you can learn a lot of biology. And so how would one answer such questions when you have a sequence? Well, there's lots of different tools, but one of the main ones that really facilitated this was developed in was actually published in 1990, and that is BLAST. So in this paper, what they showed that they had an alignment strategy of aligning sequences. But what was more important was that they connected this to DNA and protein sequence databases, so that when you now take your sequence that you might have, you paste it in this box, and you very quickly find where the sequence is found and are there any mutations associated with it. And because in the 1990s, or actually late 1980s, the scientific community had decided that as you start to publish your on sequence data, that it had to go into the public domain. So we've come to expect and know and appreciate that all the data is accessible and retrievable in the sequence community, or at least most of it. And so the reason, what made this really possible, that this was accessible and retrievable, is that we had a database where the community deposits sequence data into the public domain. Then there is a knowledge base associated with it. So if a sequence was well studied in a laboratory, that information would be added to it. And so that's a part of your knowledge base. And then you had this retrieval infrastructure. And BLAST is really the algorithm that allows them to build this particular retrieval infrastructure. So because then when you paste your sequence in here, you can find out a lot of information about that sequence. Again, if there are any questions, just feel free to holler out. But what made this really functional are two major advances. And that is, first of all, the community decided that we needed to have a uniform format. And so the .fasta file was decided to be the file format that was used in the database. And so now you had all the data in a uniform format. And the way BLAST works when you start to search the repositories is that behind the scenes you have a network of pre-computed sequence relationships. So every sequence is compared to every other sequence. And then when you paste your sequence into that box and you do a search, it searches within that sequence network space. And then it reports on every other sequence with an expectation value. And so you get this list of sequences and match scores, etc., and you figure out all the details about that sequence that we can find. And so now, if we go to mass spectrometry, when we ask the same questions for mass spectrometry, are there any analogs of these molecules that we detected via mass spectrometry? In what other data sets are they found, and what's the context of that data? And so, in sequencing, this is so common, but in mass spectrometry, there was nothing available to really truly answer those very simple questions. And so that made me a bit envious and jealous, because my whole graduate career and even my postdoc career was doing genome mining, where I tried to find molecules of new biosynthetic gene clusters, trying to uncover new chemistry or new biosynthetic chemistry associated with molecules. And the ability to find sequences in the public domain was critical to understand this information. And yet, when we start to look at mass spectrometry and try to do similar type of analyses, we essentially came up on empty-handed. And so the foundation to this solution really started in 2007. And so I'm going to give a bit of a perspective. Amazon Cloud was 1 year old at that time. Dropbox didn't exist, and it was introduced in 2007, but very limited capacity, no Google Drive, no GitHub. So really, there wasn't really many ways that we could share information. The way we actually shared data was through these hard drives and CD drives. I remember having more than 2,000 viruses on my computers because we started to share all this information via these hard drives. And we just had viruses jump from one place to another place. And so it wasn't a very efficient way to share. And so the first question that Nuno and I had is, because we started to collaborate together, is how do we more effectively share data? And we wanted something like what the sequencing community already had for 30 years, and those were functioning and working data repositories. So we have GenBank, ENA, the DDBJ, and we really felt that metabolomics also needed a functional repository. Now this is where you come into a challenge. So what comes first, the data or the repository? And the question is, why should we even share the data? Because metabolomics is already useful. And those are some of the arguments that the community made. We don't really need a repository. The other challenge that we were facing is that there was a lot of information, but there was no centralized place where we could find that information. So you have, there were two other repositories that were introduced in 2013, around the time when we introduced GMPS Massive, and those were Metabolites and Metabolomic Workbench. And then there are knowledge bases such as MassBank, NIST, HMDB, Respect, Medlin, MCCloud, MiraB. These are reference spectra of known molecules. But if you wanted to ask the question, where's this molecule found and are there any analogs found? The biggest hurdle that you had to overcome is that all the data that's stored in the public domain were in different data formats. And so if you wanted to answer these questions, you would first have to convert all this data into a uniform format. You'd have to convert some data from people's brains into a community format. And most people, and that's still true today, don't share their metabolomics data. And so you'd have to reach out to everybody and ask them if you could get their data. And most of them are not capable of providing that. And so you'd have to convert it to all the, first get all the data, secondly, convert it to a uniform format, and then develop the algorithms to be able to answer these particular questions. And so we tried to address this by building a GMPS infrastructure on top of Massive. Massive is a proteomics repository that Nina Bandera built. And we figured that we could leverage this to build GMPS on top of this. And so what is GMPS? Well, it's still evolving. And the way I think about it is that this is a data capture system, a knowledge capture system, but also an analysis ecosystem. So it's really an ecosystem that's really leveraged by the worldwide community. The heart of GMPS is really that you have an upload feature, and we take any data file or data set that we have. So somebody in Hawaii uploaded 16,000 LCMS runs from cyanobacteria, for example. We take it. If you have reference spectra, we take it. We then convert that to or we then store all this data. And then we provide data analysis features. And I will highlight some of them that we have developed so far with the community. And then there's a social component to it, feedback and predictive computing. And if you don't know what predictive computing is if you've ever ordered anything online on Amazon or eBay or similar type of online store, they often recommend other things you should be ordering. Well, that's what predictive computing is. So if you upload a data set to GMPS, we actually recommend other data sets that are related to you, for example. So the first thing that we needed to do for this to function like the way BLAST functions is that we needed to convert everything to a uniform format. And so there's maybe 30 to 60 different data formats that exist in Metabolomics. And what we do is we convert it all to MGF format. So we store both the original data that was uploaded, but also the process data. And the reason why we chose MGF, not because it's maybe the most efficient text file, is just I would have liked to have used now MZML or MCXML as the main one. But because MCXML didn't exist when we started the GMPS platform. But now, for Metabolomics, if we wanted to ask this question, where's this mass spec data found, or are there any analogs? We now have one of the key first requirements that is needed to make this possible. And that is because we now have a uniform data format for everything that's stored in our public infrastructure. The next thing is the network of sequence relationships. We needed something equivalent. And so this goes back to 2010 when I was teaching system-wide mass spec course with Nina Bandera. This is a course that I'm actually currently teaching again. And basically in this particular class, we basically had students or postdocs that would come in with their own projects. We would then collect data on it, and then we would help them analyze that data. And what we found were that there were two key projects that were actually really hard to analyze. And those were by Jeremy Watrous, who's now a faculty member here at UCSD and my former PhD student. But essentially, he was developing this nanodesi live colony profiling. And so the way nanodesi works is that you have this small solvent capillary, and then you have this breakage in that capillary and then into the mass spec inlet. And what he was able to do is raise a Petri dish to it and then look at the molecules that were there. And then he would fragment all the molecules he would see in that particular data set. Now, this was quite efficient, and so he started to get a lot of data, so hundreds of colonies that he would look at. And so he didn't have an effective way of actually dealing with that complexity of data and visualizing that information. The other one was Jen Carbush, and she was a PhD student in Lahina Alawa's lab at the Scripps Institution of Oceanography. And she was studying hopanoids in the water column. So hopanoids are almost like sterols, but they are really recalcitrants, meaning that they don't break down very easily. And they are believed to be markers of geologic times in the ocean column. And so she had samples every 50 meters in the water column. And then she had 8 reference spectra of known hopanoids. And she would manually go through each MSMS spectra. So in her case, they were long gradients. So she had 10,000 MSMS spectra, and she had 100 files that she had to go through. And she was manually comparing those 8 to every MSMS spectra in her data set. And so I thought about it, and I think we came up with a decent solution to tackle both of these problems. They were different problems, but they actually had the same solution. And a part of the inspiration of a solution came from when I visited Michael Fischbach's lab in 2010, where he showed me this picture right here. This picture is a network of comparisons of gene clusters that make small molecules. And so basically based on the domain structure of these gene clusters that make these small molecules, and you have to think about molecules like penicillin, vancomycin, etc. Based on the domain structure, you can compare and align essentially these gene clusters and build a network out of it. And so now you can see a saccharide cluster, PKS cluster, NRPS cluster, etc. And this is the foundation of his paper that he published in 2014. And this was really, that network that I'm showing you on the top here was really done by Pieter Sinnermanik and Marnix Medema, who now has his own faculty position at University of Wageningen. But I was also familiar with work that Nuno Bandeira had done while he was a graduate student. So I was on his thesis defense. I was part of his thesis defense committee. And what Nuno had developed was this way to essentially de novo protein sequences based on spectral network analysis. So in 2007, if you were to do de novo sequencing of peptides or proteins, you would make errors in about 1 out of every 4 amino acids. And so what Nuno did is decided, well, what if you improve that by digesting a protein with as many proteases as possible? So you get these overlapping peptides and then start to sequence these relationships. And that essentially is through building a network. And this improved the de novo sequencing from 1 out of 4 amino acids being erroneous to 1 out of 10 being erroneous. And he has since improved this to 1 out of 100 being erroneous. And but it was a major advancement. But I distinctly remember when I was on this thesis committee, this picture right here. And I also remember him saying that he first filters the data for amino acid sequences. So if you have an MSMS spectra, you get these mass shifts associated with each of these amino acids when you look at peptides. And so he first cleaned up the data by filtering for amino acids. And so I was sitting in the class with him, and I basically said to him, could you take this data set that I have for you, and can you turn off the amino acid filter in your code that you published in 2007? And I believe that this will work for looking at relationships between molecules, all molecules. And he argued with me so heavily that it will never work, and there's a variety of different reasons. But if you've ever worked with me and I get excited about something, I'm very persistent, and I basically don't stop until you prove me sort of wrong. And so he finally broke down and actually coded it while he was sitting in class. And so what he coded was the ability to merge all spectra, which was a clustering algorithm that had just been developed by Pavel Pessner as well, called MS Cluster. And so first you merge all identical spectra. And then what I wanted him to do is say, okay, I want you to look for similarity of these spectra so that we can potentially find molecules that are related. And I'm just hypothetically showing an example here of caffeine would then be connected to theobromine, because the only difference is a methyl group right here, or paroxetine, where a methyl group would be right here. And so this turned out that this worked quite well. This was maybe the second or third molecular network that we ever created. And Nuno was like, it doesn't work very well. And whereas when I looked at this, my jaw dropped and I was like, this is phenomenal. This is amazing. And, you know, there's just sometimes different perspectives that people have. But to me, this really transformed the way I do mass spectrometry, the way many people do mass spectrometry nowadays. There are these very early pictures of it. Why did I think this was amazing? Well, if you look at closer, you start to see these subnetworks. You start to see these subnetworks. Okay, but so I was really excited, and it was working, in my opinion. And so I went to try to publish it. It was rejected 8 times by 5 journals. And yet I felt like this was a similar advance to what allowed text mining and eventually developed Google, for example. So what I did was I contacted Gerald Meinwald. He unfortunately passed away, and told him this is the most important paper that I've ever produced in my scientific career so far, which was fairly young. I was an assistant professor, but I can't get it published, and I really am struggling. And that was because only 1 out of 5 people that looked at these networks understood the implications. Fortunately, when it was published, Roberta Coulter wrote a phenomenal commentary on this particular paper. And this really set the stage of people paying attention to this particular approach. Now, we then expanded it into using networking to enable dereplication or variable dereplication. Application in the metabolomics community is also the equivalent of finding known unknowns, whereas dereplication is used in the natural product community and finding known unknowns used in the metabolomics community. So what is dereplication or variable dereplication? Well, the basic principle behind dereplication is that if you have a panel of reference spectra of known molecules, that you can now match those MS spectra up to your data set. And so you can see how sparsely populated. I've shown this sort of fictitious mapping of reference spectra. And that was really the reality. When we started this, on average, we had 1.9% of the data had annotations. If you went into the ocean environment, it was 0.1% or even less. If you went to human data sets, it was maybe 5%. And so it was data set dependent, but there were very few annotations. But now we can even propagate some of our annotations. So in red, we have 2 examples of molecules that had matches to reference spectra. And so these are the drugs diphenhydramine and the glucuronylated version of diphenhydramine. However, then if you look adjacent to it, we have 2 additional nodes. One of them is increased by 16 Dalton, and the other one is increased by 56 Dalton. And so me as a chemist, when I see 15.996 Dalton, that screams oxygen to me. I mean, we can look at the periodic table, and then we can start to propose where that modification might be. And so this would be the diphenhydramine N oxide. And then the 56 Dalton is actually the acylated version where this nitrogen is acylated. So we've used this and leveraged this in many different examples. Here's one recent example that I'm really personally really excited about it, because I was the one that discovered this as I was looking at the data. But here we have basically a small subnetwork where we have GMPS library hits. These three now have GMPS library hits. But at the time, only this one had a library hit. And this was a match to glycolic acid, which is one of the bile acids that sits in our guts, for example. And if you look at the MSMS spectra, now we can see this glycine shift because we see the fragmentation over at this amide position. And so we see it on both ends. And so now if we look at these neighboring nodes that we did not know at the time, what we can see is that these mass shifts correspond to phenylalanine or tyrosine instead. And so this is how you can start to do variable replication and really propagate some of the original annotations that you can get and learn a little bit more about the molecular details of these unknown molecules. And so we then built this infrastructure for the community. And this was written with 127 authors, and we have more than 200,000 accessions a month now. One of the trickiest things was, so to be able to do this type of analysis, we needed to have reference spectra. So the community had some reference spectra available, but these reference spectra are really biased towards commercially available compounds and compounds that are much more widely studied. And so primary metabolism is much more widely studied. And so secondary metabolism is very infrequently studied. And thus the reference libraries that exist in the public domain are devoid of reference spectra of those less accessible molecules. So a lot of the microbiome molecules, microbiome-derived molecules, we just don't have reference spectra for them. And so what we did within the infrastructure is we gave the ability to people to upload their entire data sets, but then also make annotations of those data sets. And as soon as you annotate an MSMS spectra, that information is retained. So it could be an annotation of this, I believe this is a sugar, that's fine. Somebody else may come in and say, this is azithromycin. We will retain all those pieces of information. And then if that MSMS spectra is found in any other data set, we now send an email to the people who subscribe to those data sets. So you get this worldwide learning about the data and really create a connected world in a data-centric manner. And so this has been quite successful now. And you can see sort of the growth of reference spectra that the community has provided. So 140,000 reference spectra. It has increased the annotation rate from 1.9% to about 5%. Right now, we're actually seeing a decline in annotation rates, and partly is because the amount of data that's growing in GMPS massive is surpassing the rate of library spectra deposition, and the number of unique spectra that are found that is deposited in the database is growing compared to the number of known molecules and with their reference spectra. But nonetheless, there's a lot more information available now than it existed when we started early on. So that's pretty exciting. We've also brought in all third-party libraries that we are allowed to make publicly available within the GMPS environment. So you can go to one place and search all those. The other thing that happened was that GMPS became a recommended data repository, so GMPS massive, and we can provide both an accession number or a DOI if the author chooses to. And so what are we currently trying to do? So some of the things that we're currently trying to do is allowing other data types to be analyzed as well. So far, it was only done on LCMS, and really LCMS where you had a parent mass and MSMS fragmentation data. And so we're starting to introduce GCMS capabilities within the infrastructure. And then I'm going to briefly highlight an example of ion identity molecular networking as a new tool that's being developed as well. So the GCMS platform was just published, but what this is really about is one of the challenges that we face with GCMS data analysis for the GMPS environment is that all of the deconvolution tools that existed for the community, they didn't scale. So they either couldn't handle the number of files or they're too slow. And so the other challenge that we were facing is that if you wanted to analyze GCMS data, you needed to know a lot about the instrument on which it was collecting. And you need to know a lot and be a true expert at interpreting the GCMS data first before you can do deconvolution, because you need to understand the amount of trailing, you need to understand details about the retention shifts. There are lots of little details that you need to know normally when you do deconvolution. So we wanted to have an approach where you can just put in your data and it does auto deconvolution for you. And then you take those MSMS spectra that then come from GCMS. You can then use those to search libraries or do molecular networking with. And so the big advance really came from a collaboration with Kirill Vesakov at Imperial College. He developed an approach called MS Hub. And MS Hub is basically, it's a really pretty amazing tool. But basically what it does not deconvolution at a single file level. No, it does deconvolution across the entire data set. And so what it does is you first find ions and you find it across the entire data set. So you get a frequency across of this particular ion across the entire data set. And then you do Fourier transform on that and you just do signal matching. And you match the frequencies. And in principle, the only ions that really match to those frequencies are the ones that have the exact same behavior. So now you start to get good fragmentation data by leveraging the entire data set. And so that allows you to essentially get a value, which we call a balance score, that basically describes the quality of that feature as it's represented in the entire data set. And so once it's run, you don't have to do any input parameters. Once it's done running, the only decision you have to make is what level of quality you like to include in your subsequent analysis of library annotations or molecular network analysis. And so molecular network analysis for GCMS is very new. But what is really nice is that with GCMS, it really groups really nicely according to these different classes of molecules. And so let me zoom in over here where we start to annotate some of these molecules. In this case, you'll start to see that there are a lot of fatty acids represented here. And so here we have one node where there's no spectral match to the library, but because the MSMS spectra is so similar, we can probably predict that this is a fatty acid as well. And so this is one way that molecular networking can help you in GCMS annotation. And so the next example of a recent tool that we're introducing is ion identity molecular networking. And I'm just going to show you how ion identity networking really works is through native electrospray-based metabolomics, another strategy that we're starting to introduce. One of the things that the metabolomics community doesn't really pay attention to significantly enough is the role of metals or metal interactions with small molecules. So some fatty acids, for example, have specific functions in biology, but they will not be functional if they don't have their potassium counterpart. Similarly, if there are ionophores like siderophores, they are designed to pick up iron. There's also molecules like calcium-dependent antibiotics. These are antibiotics, and without calcium, they are not functional. And so there's a lot of small molecules that probably require a metal counterpart. And but how do we discover what the candidate metal counterparts are? And so we had an idea, and basically the idea was, can we overcome the limitations of typical mass spec conditions? Because if you do chromatography by mass spec, most often we add some sort of acid to it. It really improves the peak shape when you do HPLC or UPLC chromatography. But if you use acidic conditions, by and large, not always, by and large, it does not preserve the metal interactions of these small molecules. And so what we decided to do is basically take an HPLC column and then just do standard chromatography. So you have your flow of your solvent here that's at low pH. But what we decided to do was we decided to introduce a base that then adjusts the pH to whatever you'd like it to be. So it could be pH 5, it could be pH 8, it could be pH 10, whatever you choose it. And then we can infuse our metal salts. And so what that allows us to do, it allows us to then bind to these molecules where it would otherwise prohibitively prohibit binding of these metals to these molecules. And then those would be infused into the mass spectrometer, which is great. So if you take a mixture of known molecules that bind iron, for example, such as the ones that are shown right here, and you perform molecular networking, what you get is you get these nodes that are bound to iron as separate adducts versus the apo ones. They're not connected. And that is because the fragmentation behavior of these molecules change when metals are bound. This is not always 100% the case, but certainly if they're ionophores or siderophores, as the ones that you see here, that is certainly changing the fragmentation behavior. And so imagine if this was a complex pool from, let's say, dissolved organic matter in the ocean or a fecal sample, and you would want to find a signal that would have a metal bound, you would actually have a really hard time to find those metals. So iron does have an isotopic signature, but other metals such as zinc do not. And so you couldn't even use isotopic signatures. And so what we decided to do is we decided to both leverage feature-based molecular networking and ion identity molecular networking. Feature-based molecular networking is basically allows us to connect the feature or area under the curve of the peaks with the fragmentation data. And this was really key to allow ion identity networking to take place. So it took us from 2017 to 2020 to really finish up a good form of feature-based molecular networking. But once we did, it was possible to do ion identity networking. And the way ion identity networking works is that if you have a feature either within the same data file or among different data files, if you have the same feature shape, you should be able to find it. So in ion identity networking, if you have protonated version, sodiated, in-source fragment, and they all loot at the same time, and they have the same peak shape, it allows you to find those as well. And so we thought we could leverage this in ion identity networking. And so we did one run without metal addition, but pH adjustment, and then one run with metal addition and pH adjustment. And then we do retention and peak shape correlation. And then we do filtering. And so basically we look for specific mass shifts. So iron would have a 53 Dalton shifts because when you have a molecule that complexes with iron, you have to remove two protons and then really 3 protons and then add the iron to it. and then another proton so that it's charged. And so when we do this in this particular project, what you can then see is you can start to see these additional connections between the apo form, the unbound form of the molecules, and the iron adduct. And just to show you, so here we have the protonated form of enterobactin. Here's the sodiated form of enterobactin, and these self loops show that even those are normally not connected in a molecular network. But now with ion identity networking, we can link those. But in addition, with the iron adduct, we can now see that those are connected. And so this is really easy, allows you to easily discover different types of metal adducts. And we've now used this to actually uncover how E. coli Nissal does zinc absorption to prevent salmonella infections, for example. So the future, I think, so now I've shown you 2 examples of what's currently going on. There are 1,000 other things going on. And I'm happy to discuss a lot of these, but the future is similarly bright. It's really, the field is transitioning into sort of a data science field. And really what we're trying to do is create data table where data is interconnected when they're generated by different approaches. And these are the 4 things that I would like to really cover a little bit. One of those is reference-based metabolomics. I think this is another example that is going to transform how we think about leveraging data that has been collected in metabolomics and that have been deposited in the public domain. We're beginning to create new capabilities for data scientists, and really this is, so you now start to get non-mass spec people doing metabolomics analysis. We're making data discoverable and really trying to connect the world's mass spec data. And really what we're trying to do is democratization of science, and particularly metabolomic science. And so if we're going to go, first I want to highlight the democratization of science, and that is linking to other resources, but also easier to begin using the platform. So the first part of democratization of the science is that all of this infrastructure is free to access, to use, and download. And so far we've covered the cost for this. And I think this is really important. There are lots of laboratories and lots of places that don't have the resources that we have. However, by other people using the infrastructure and contributing back to this infrastructure, we can make it more powerful. And that is allowing us to get good grants, good support, that allow us to make this infrastructure free. We've made extensive modular tutorials. So if you go to the documentation page that you can see in the banner when you go to gmps.ucsd.edu, we have lots of training modules. So anybody who doesn't have time to go to workshops, for example, they can learn on their own. We also give a lot of workshops. We don't only give it in English, we've given it in Spanish, Portuguese, Korean, Russian. And that is because even though English is the main scientific language, by not giving it only in English, it removes another barrier for people to start beginning to use the infrastructure. We also have office hours, and there's a forum where anybody can ask questions and the community will provide answers on this. So the other thing that we're trying to do is making it easier to begin using the platform. We realize that the barrier to start using a platform like this is pretty high. And so we are starting to build versions where basically you can drag and drop files to start depositing data, to convert data from one form to another, and to perform certain types of analyses. And this is work in progress, but this will really improve the ability and accessibility to the community. Now, the democratization, I think, has worked quite well so far. Users from 168 countries have now used the GMPS infrastructure. This is just an example in the past week in terms of number of people who have submitted jobs in the past week. So you can see it's quite a large number. And even as I was preparing this presentation, I looked up who was using it at that time. And you can start to see countries like Philippines, Thailand, and Zimbabwe. Those are not typical countries that you would think would use an infrastructure like this, or they have fewer resources to do high-level science. And this is allowing those kind of countries to also do high-level science. So the next part I want to highlight is really creating new capabilities for data science and making data discoverable. And so we're really creating and partnering up with a lot of different community resources. There's the latest one, and I think some of the people who are on this Zoom are probably a part of this effort. But basically, community resource that is both paired genomic and metabolomic data mining, where you link metabolomics to the sequence information. And this is really spearheaded by Michelle Shorn, Justin van der Hoofd, and Marnix Medema. Then we have the redo, which is a metadata curation strategy, so that in the future you can essentially do text mining as well to find all the data sets that have that information. So for example, if you want to find data sets of women under the age of 2, now it's a couple of clicks on the button. Or if you want to find environmental samples in specific longitudes and latitudes, this is now becoming possible through the redo mechanism. Cheetah is an integrated analysis platform. This paper was published in 2019. It has more than 1,500 citations at the moment, but it's basically an infrastructure that allows you to do quantitative analysis. So any statistical approaches, and we're building really nice infrastructure to relate mass spec information to sequence information, and similar to QIIME 2. And we have just submitted a grant with Metabolites, which is the European Metabolomics Repository, trying to link metabolites and GMPS and really to improve the quality of metabolomics annotations. And I'm just going to highlight 2 examples of the kind of things that become possible. So because we had linked GMPS and QIIME, we can now do microbiome analysis and really develop new algorithms to improve the analysis that can be done. And so here is an MMVec plot, for example, which is really a bilog linear multinomial regression analysis. But here, each dot is a molecule. The gradient, color gradient, is a high-fat diet versus low-fat diet. And then the arrows are the microbes that are most strongly associated with these molecules in these directions where these arrows are pointing. And so if we look at these phenylalanine conjugated acids that I mentioned earlier, what we found was that they were associated with Clostridialis. And so based on that, our hypothesis was that microbes are making these molecules. And we could verify this in a Petri dish when we cultured different Clostridia, and we simply looked for the production of the phenylalanine cholic acid. And so now this is an example of how you can take a very complex data set, a human data set, and begin to tease apart some of the relationships. But the amazing thing that we can start to begin doing now, and I'll finish up really quickly here, is that we can start to do co-analysis of large amounts of data. So here we're looking at a data set that has about 200,000 samples of skin, fecal, and oral data. And then we're looking at one person from birth to the age of about 2.5 years. And you can see the migration of these molecular profiles to adulthood. And so it's these kind of visualizations that sound like that are very hard to do, but because we're integrating with Cheetah, they're actually very easy to do. And literally we have high school students coming in that are capable of generating these kind of analysis plots. But because we have this infrastructure now, it's a uniform format, having a master molecular network, there's so many things that we can do. We have 50+ tools, but one of the most important ones that we have recently introduced is the mass search, which is the equivalent of a blast search, but now for molecules. And so let's say this was one of the original questions that I posed. Is the work in an animal model translational to humans? This would be a very difficult question to answer. But now it's no longer difficult, because what we do is we paste the precursor mass here, the fragment masses with their relative intensities, and then we just hit go and simply ask the question, are these matches found in any human data sets? And in terms of the bile acids that we discovered in mice, we indeed were able to find that they were found in human data sets. So it was really translational to people. What we knew as of January 19, we have a new feature, is that we can now also search all the analogs. So if you search with one molecule, you can now all of a sudden find all the related analogs within this infrastructure as well. So this is a pretty exciting introduction. And so finally, what I want to leave you with is this concept of reference-based metabolomics. And so in traditional metabolomics analysis, untargeted metabolomics, you fragment all the ions that you see. So you get these fragment spectra, and you match them up to a reference library, so you get some annotations. Now, the average annotation rate in humans is now about 10%. And so that means that 90% of the data, we don't have any information. So what we decided to do is basically take reference data. So if you have reference data, and this is focused on food, but it could be of personal care products, it could be of anything else that you can conceive of. But let's say you have reference data of wine, citrus, honey, and basically you run a standard metabolomics experiment on each one of these molecules, or each one of these samples. And so you get about 2,000, let's say, MSMS spectra. But instead of having an annotation, we now have an annotation that this is a wine molecule, that this is a citrus molecule, or that this is found in honey. And so now we can co-network these two and really begin to propagate some of these annotations. And so here in black, we would have a node that's found in the, let's say, a human data set. And it match up to a food-derived molecules. And so now you have this connectivity, even if it's metabolized. And so all you now need to do is do a readout, in this case, of food, of the origin of the spectra, and sort of create summaries. And so we can do that for every person now, if we look at diet associated with people. And we can do statistics on this. And so here we have each dot is a person, and we just do PCA analysis of all the food-derived information that we have. And then if you have microbiome data or you have disease activity associated with it, you can now begin to associate this with disease or microbial communities by doing linear regression analysis. So in this case, what we see is that fleshy fruit and vegetables are associated with higher microbial diversity, and that meat is associated with worsened disease severity in IBD. And we see similar trends in Alzheimer's disease, where if you have more legumes in your diet versus more soda in your diet, you have, if you have more soda, you have an increase in dementia. You have increase in your cognitive, negative cognitive index, and your legumes are more associated with improved forgetfulness, i.e. Better memory. And so these are the kind of relationships that we can now build. But what I want to leave you with is with some take-home messages. First, envy is generally a negative thing, but sometimes envy can drive you to do things that you didn't think was possible. When I was starting as a faculty member, I never thought we would be developing these kind of tools and that the community would pick these up. I would always encourage you to work with others, share your data and knowledge, because that will really democratize science. We often many labs work in silos. I think silos slow down the science. And so the sooner you share, the faster the science is going to go. And it's going to be a matter of publishing 1 paper versus 10 papers in the same amount of time. That's the scale that if you work in silos or not work in silos. There's a need to connect different worldwide data, as I indicated. But again, I really want to highlight to you, share your data. I think this is really, really important. And then finally, lots of people to thank. This was the community that built the GMPS infrastructure. This was the first, all the authors on the GMPS paper. These were the people on the MAS paper, the Chine paper. So these are really not done in silos. These are really major community efforts. And you can see the impact that it's beginning to have. And so, of course, I need to acknowledge the funding, and then, but most of all, this amazing team. So I get to speak about the science, but the people who really do the work are the ones that you see in this particular picture, and some of the ones that are not pictured yet. But it's just I love going to work because of these creative minds. These creative minds do things often without me telling them to do things. And I'm super impressed with their accomplishments. And then finally, I want to thank you for your attention, and I'll be happy to answer any questions.